Good afternoon, dear students. Now, today also we are going to continue with the topic, that is electrophoresis, that we studied in the last week also. So, we have studied what is electrophoresis, what are the different types of electrophoresis, and also one of the type of gel electrophoresis, that is agarose gel electrophoresis. We studied in the last lecture. Now, today we are going to discuss about SDSPAGE. Before we understand what SDSPAGE is, let us understand what are solubilizers that are used before performing SDSPAGE. Now, most of the proteins are multimeric. That means they are made up of one or more subunits or one or more polypeptide chains. The one or more polypeptide chains present in a protein are held together by hydrogen bonding, disulfide bridges, hydrophobic associations. And these subunits need to be separated from each other before performing electrophoresis. Why? Because in their native form, proteins fold into a variety of shape and some amount of and become compact and some are elongated. Okay, so in their native form, when the different polypeptide chains are folded, they are held together by hydrogen bond or hydrophobic association or disulfide bridges, they have a particular shape. Some may have a globular or compact shape, some may have elongated shape. So, the rate of migration, when you are performing electrophoresis, the rate of migration of the native protein through the gel is therefore a reflection of their relative compactness, and we get a less accurate result. And hence, denaturation of proteins or separation of subunits of protein will reflect mobility depending upon true charge to mass ratio, and that's why the subunits need to be separated. Okay, and also if you want to know that the multimeric protein is made up of how many subunits, then you need to separate the subunit. So to separate the subunit, we use destabilizers. Okay, we use destabilizers. So the units are separated from each other by destabilizers by breaking the various bonds, and collectively the destabilizers are known as solubilizers. So what are the different solubilizers that we use to break the protein into their respective subunits? Okay, number one, urea. Urea is used to break hydrogen bonds that are present in proteins reversibly. The concentration in which urea is used is 3 to 12 molar. Okay, so the first solubilizer is urea, used to break hydrogen bonds reversibly in a concentration of 3 to 12 molar. The second solubilizer is sodium dodecyl sulfate. Okay, sodium dodecyl sulfate is a strong anionic detergent. It is a strong anionic detergent. It acts as a hydrophobic bond breaker. Now, SDS binds to the hydrophobic region of the protein and gives the protein a negative charge, complete negative charge to the denatured polypeptide. See, whenever a protein is treated with sodium dodecyl sulfate, SDS, in short, being a strong anionic detergent, will open the protein. It will make the protein into a thread. Even if it is entangled, it can break the okay, it will convert the protein into a thread, into a long thread, and swap all the charges that are present on protein, and the protein will ultimately get a negative charge. Okay, so all in all, sodium dodecyl sulfate, a strong anionic detergent, okay, you can say, destabilizes the protein. Okay, converts its compact structure into a thread like structure, into a long tube like structure, and gives complete negative charge to the protein. It overshadows all the other charges, converts the protein into a negatively charged, you can say, thread. If you want cationic detergent, sometimes we require a cationic detergent where the molecule needs to be offered a positive charge. At that time, we use CTAB. 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 Okay, cetyl trimethyl ammonium bromide. Cetyl trimethyl ammonium bromide. CTAB. It is a strong cationic detergent. Next in the series of solubilizers is beta mercaptoethanol. Beta mercaptoethanol is required to break disulfide linkages. Okay, beta mercaptoethanol treatment is followed by iodoacetate treatment so that the separated subunits do not reassociate. Okay, so solubilizers are urea to break hydrogen bond, sodium dodecyl sulfate to destabilize the hydrophobic attraction and convert the protein into negatively charged molecule, cetyl trimethylammonium benzaldehyde to as a cationic detergent, and the third is beta mercaptoethanol to break the disulfide bridges. But whenever we use beta mercaptoethanol, this treatment is to be followed with iodoacetate treatment to prevent reassociation of the protein threads. Now, let us study electrophoresis of proteins by sodium dodecyl sulfate. Now, you can see in this diagram, there are hydrophobic interactions or hydrophobic areas where the SDS will bind and convert the folded protein into a straight rod-like, thread-like protein and offer all negative charge. So, it's a very strong, you can say, detergent. Now, SDSPAGE. SDSPAGE is a method for electrophoresis of protein, separation of protein, isolation of protein. SDS means sodium dodecyl sulfate, which is a strong anionic detergent. PAGE means polyacrylamide gel electrophoresis. So, sodium dodecyl sulfate polyacrylamide gel electrophoresis is mostly used for separation of proteins according to their size. It is one of the most powerful techniques to separate proteins on the basis of their molecular weight. The technique uses anionic detergent sodium dodecyl sulfate, which dissociates the protein into the individual polypeptide subunit and gives the protein a uniform negative charge along the denatured polypeptide. Okay, along the denatured polypeptide, the whole protein, as you can see here, will get a negative charge. You can see here in the second figure, all negatively charged molecule after treatment with SDS. SDS also performs one important work. It forces the polypeptide to extend their conformations to achieve similar charge to mass ratio. So, charge to mass ratio is same. SDS treatment therefore eliminates the effect of difference in shape so that the whole proteins get separated only or migrate only on the basis of their molecular mass. Right. So, molecular mass is the sole determinant of migration of proteins during electrophoresis. When the denatured proteins are loaded at the cathode end, why do we load it at cathode end? Because it is negatively charged. Of the cathode end of the electrophoretic tank having polyacrylamide gel as the supporting media and subjected to electric field, then we get clear bands of protein arranged in decreasing order of their molecular mass from cathode to anode. Small protein molecules will move faster towards anode. Large protein molecules will move slower towards anode. So, from anode to cathode, we get all protein bands arranged. The rate of movement is influenced by the pore size of the gel and the electric field also. So, in SDSPAGE, we use vertical gel electrophoresis, means top to bottom. Top is cathode, bottom is anode. So, from top, we load the protein sample and it moves towards the bottom of the gel. Although SDSPAGE is used to separate proteins, we can use it to separate DNA and RNA. In most of the electrophoresis unit, the gel is mounted between two buffer chambers. As I said, upper chamber and lower chamber. Upper chamber is cathode, lower chamber is anode. Okay, and between the upper and lower chamber, what is connecting the buffer in the upper and lower chamber? The gel. Okay, so gel separates the two electrode. So only electrical connection between the two chamber is through the gel. As you can see the apparatus here, vertical gel electrophoresis apparatus. Sample is loaded into the gel. You can see wells are created and in the wells we have loaded the sample which is in blue color, and the upper chamber is having buffer in it which is cathode. In between there is a gel and in the lower chamber there is buffer, it is the anode. So proteins will move from upper chamber into the lower chamber. Oh, right. You can see here more clearly. So upper chamber, negative electrode or cathode, stacking gel. In the upper chamber, we take stacking gel to some extent. Yes, you can say 1/3 is stacking gel and then it is running gel. Running gel where the proteins get properly separated on the basis of their molecular size, you can say, or molecular weight. and the gel is casted between two plates. There are two acrylic plates which are clipped, where we place the spacers for depending upon the thickness of the gel you want. We plate spacers between two plates, clip them, and then we pour the running gel, and over the running gel we place the stacking gel. Okay, what is running gel, stacking gel? I'll explain you. Running gel is the point, it is 2/3, and say 1/3 is stacking gel. In the stacking gel we have created the wells, we load the sample. See, every time the protein sample is to be mixed with a tracking dye like bromophenol blue, and any agent which will make the sample heavy so that it sinks into the gel. I explained you in agarose gel electrophoresis also. So the protein sample is made ready by mixing it with you can see it contains buffer also, and number two, it contains any agent which will give more, make it more dense or heavy, like fecol or sucrose or glycerol, and a tracking dye. Okay, so and then it is loaded into the gel. So it moves through the stacking gel where the pore size is small, and then the where the pore size is larger, and running gel having small pore size, and then the protein molecules get separated depending upon their size. This type of electrophoresis is called as discontinuous gel electrophoresis, because we have used two different types of gels, stacking gel and running gel, where two different systems of gels are present in the electrophoretic tank, physically placed one over the other. We call it a discontinuous system of gel. There are two types of gel, resolving gel, that means the running gel, and above it we have a stacking gel. Running gel is used, also called as separating gel, resolving gel. The separating gel consists of 2/3 of the length of the gel plate, prepared by using 5 to 10% of acrylamide. Pore size in this gel, which is formed after polyacrylamide is cross-linked, are numerous and small in diameter, which imparts a proper sieving effect to separate the proteins on the basis of their molecular size. So resolving gel or running gel, you can say, occupies or constitutes 2/3 of the length of the gel. It contains 5 to 10% acrylamide, which after polymerization gets converted into polyacrylamide. There are numerous small pores. Okay, the diameter of the pore is very small, and it imparts a proper sieving effect. Sieving means what? छानने को, जिसको हिंदी में जिसको हम छानना बोलते हैं. Okay, so a sieving effect, right? So small molecules move faster, large molecules move very slowly, or they get retarded. So proper separation on the basis of molecular mass will occur, molecular size. The stacking gel, above the running gel we place a 1/3 layer of stacking gel. Stacking gel is poured on the top of the running gel, and a gel comb is introduced into the stacking gel so that wells are formed in the upper layer of the gel, and it constitutes 1/3 of the gel plate. Percentage of acrylamide depending upon the size of the protein. So actually the pore size is larger in stacking gel, so we have to use less of you can say acrylamide. The smaller the known weight, the higher the percentage that should be used. Generally, the percentage of acrylamide in stacking gel is 2 to 5%. So, pore size is very large. It is highly porous and devoid of molecular sieving action. So, there is no molecular sieving action. It actually allows all the protein molecules to reach as a band up to the separating gel. Okay. Now, once the sample is loaded, see, the purpose of the stacking gel is to concentrate the protein sample into sharp band before it enters the main separating gel. Okay. So purpose of stacking gel is to concentrate the protein. The proteins move very fast through the stacking gel and concentrate as a sharp band before it enters into the separating gel. This is achieved by utilizing difference in ionic strength and pH between the electrophoresis buffer and the stacking gel, and involves a phenomena known as isotachophoresis. See, the you can say pH of the buffer that is used in the upper tank, or that is used to prepare stacking gel is different, and the pH of the buffer used to prepare running gel is different. See, the pH of the stacking gel is 6.8 and the pH of the separating gel is 8.8. Okay, so the pH is 2 less, 2 number 2, like this, 6.8 in the stacking and 8.8 in the running. Okay, so the pH is more acidic in the stacking gel than in the separating or running gel. Okay, so here we use different kinds of buffers also. So two types of buffers are used in SDSPAGE. The lower chamber, which has the running gel, has amine buffer. It is adjusted by using HCl. The upper reservoir, which has stacking gel having pH 6.8, also use TIE buffer, but a tris buffer actually, but its pH is slightly above the running gel and is adjusted with glycine instead of HCl. When you perform electrophoresis of proteins, as I told you, the most important technique method is SDSPAGE. Okay. So protein sample is first boiled for 5 minutes with buffer solution containing sodium dodecyl sulfate and beta mercaptoethanol. See, sodium dodecyl sulfate, both the proteins get denatured and open into a rod shaped structure. Because you have boiled the protein with SDS, the protein will acquire all negative charge. It will get converted into a thread. Beta mercaptoethanol will break all the disulfide bridges. Okay, so once your protein sample is ready with negative charge and converted into a rod shaped molecule, you mix the protein with bromophenol blue and a tracking dye. Bromophenol blue is a tracking dye, and you add glycerol or sucrose or fecol to make it heavy. Before the sample is loaded into the separating gel, a stacking gel you to pour, as I told you. So the sample is actually loaded in the stacking gel. Okay, now once you load the protein into the stacking gel, current is switched on. Negatively charged protein molecules now continue to move towards the anode as they pass through the separating gel. First they cross the stacking gel, concentrate as a band on the running gel, and then they start moving in the separating or running gel owing to the molecular sieving properties of the gel. Okay, now the dye which you have put is a very small molecule. Okay, so it moves very fast. Being a small molecule, bromophenol dye is totally unretarded, and therefore it indicates the electrophoresis front. When the dye reaches the bottom of the gel, the current is switched off. Gel between the two glass plate is removed. It is shaken in an appropriate staining solution. Okay, so you shake it in an appropriate stain solution like Comasi blue, Comasi brilliant blue and all, for a few hours, and then washed and destain overnight. So you have to place it in a destaining solution overnight. The destaining solution removes the unbound background dye from the gel, leaving the stained proteins visible as blue bands on a clear background. Okay, so overnight destaining is important to remove the blue color from the rest of the gel. The blue color should be only with the protein bands. Okay, and the blue color bands are observed under, you can observe it against clear background, no need of UV rays also. Right, so now you can see in this diagram, at cathode we have wells, mixture of protein mixed with gel loading dye, that is tracking dye, bromophenol blue, and sucrose or fecol is loaded. It first moves through the stacking gel and then moves through the running gel towards the anode depending upon their size. Okay, now this is the technique. So here you have an upper chamber and you have a lower chamber. See, the equipment consists of two compartments, lower chamber and upper chamber. There are two components also. One is buffer reservoir and a direct current DC power supply, we call it. The buffer reservoir has upper and lower buffer reservoir connected by gel. Platinum electrodes are positioned in the reservoir. The whole assembly is covered with a perplexed shield. You have to cover it with the shield, so there is no evaporation effect is avoided. The sample is prepared in a high density glycerol or fecol to mix, which prevents mixing with the upper reservoir buffer. Tracking dye is added. Okay, the extent of migration of the dye gives an electrophoretic front. The pH is usually fixed at 9, which gives a negative charge to the macromolecule. Anode is placed in the lower buffer, cathode is placed in the upper buffer, and you can see the proteins move first through the stacking gel and then through the running gel. So it's a kind of discontinuous electrophoresis. Okay. Here the proteins are separated on the basis of their molecular size, and longer protein molecules remain at the top end and shorter protein molecules travel very fast and move towards the anode. So once you remove the gel, stain it and then observe it, you can see clearly proteins in the form of bands, blue colored bands. What are the applications? It is used for estimation of molecular weight of proteins and nucleic acids, determination of subunit structure of the protein, purification of the isolated proteins, and monitoring the changes of protein content in the body fluid. So that is all about polyacrylamide gel electrophoresis, and we will stop here.